Thank you everybody for attending today's webinar. As Katie said, my name is Reynolds, and I am a technical sales specialist with DNASTAR. And today I'm going to be going over the Megaline application as well as the GeneQuest application of the Lasergene Core Suite. And I'll be focusing in the Megaline module on how to enter sequences in for alignment, doing a multiple alignment, looking at some phylogeny information, and then performing a pairwise alignment. For the GeneQuest module, I'm planning on going over some gene discovery and annotation tools, as well as a brief presentation of how to align exons to a reference. And with that said, I'd like to go ahead and introduce you to the software. So let me share my desktop. So with all DNASTAR workflows, the easiest place to begin is through our DNASTAR Navigator. And the Navigator, in addition to being a useful tool to launch the various applications, it also gives additional information on what each application does so that you can easily find resources on how to perform your particular project workflows. For instance, if I hover over the Megaline application, it gives me a description of what Megaline does, as well as links to our website where you can find videos, get frequently asked questions answered, as well as download our tutorials and some data sets for sample data. And to begin, we're going to go ahead and launch the Megaline application. Now, when you begin working in Megaline, the first thing you want to do is enter in your sequences into the program. Megaline can use sequences in a variety of file formats or directly downloading them from NCBI's Blaster Entrée database. I'm going to begin by showing you how you can bring sequences into the program directly from NCBI's Entrée search, which is a very easy and quick way to download sequences from NCBI when you know the unique identifier for the sequence you're interested in. To do that, you simply go into the File, enter Entrée Sequence, and that brings up our search menu. I'm going to go ahead and enter in the particular sequence that I'm going to search for. And then I'm going to select the appropriate database. In this case, this is nucleotide data, so I'm going to select nucleotide and click OK. Now what it's done is it's actually sent that request to NCBI's Entrée database and found that particular sequence using the unique identifier and brought it back for me to save. I'm going to select the folder that I want to save this in, click Save, and I've just entered in my sequence. Now the sequence that we just added to the program is the entire MIP-A gene for Aspergillus, complete with introns and 5' and 3' non-coding regions. However, if we just wanted to utilize the coding regions of a polypeptide associated with this particular gene, because we downloaded this sequence directly from NCBI, it includes with it all of the annotations and features that are associated with this sequence. So one of the features that Megaline can do is utilize this information to present only the MIP-A polypeptide. Simply go to the select the sequence that we want to adjust. We're going to go to the options, and we're going to choose Set Sequence Limits by Feature Table. Now here it's brought up a list of all of the annotations that are associated with this particular file, and I'm just going to go to the MIP-A coding sequence. Below you can see all of the sequences that are part of the coding region listed. So if we click OK, we can now see that only the coding region of this particular sequence has been selected. We can verify that with the start codon in the beginning as well as the stop codon at the end. From here, if I wanted to view this as a protein, I could simply choose the Show as Protein button, and it will translate that into our polypeptide sequence. The next thing that I want to do is, if I wanted to enter in additional sequences from NCBI using BLAST, I can simply select the region that I'd like to BLAST, go to the Net Search option, and choose BLAST Selection. I'm going to choose the database that I want to use. By default, it knows that this is a protein sequence, so it's selected the BLAST-P database. However, I can also use the tBLAST-N database if I wanted to translate this into a nucleotide and get my results back as DNA. I can select the database. In this case, I'm going to use the NR database. And once I've done that, I'm going to click OK. Now again, the program is currently sending this BLAST query directly to NCBI, and will bring the results back into this window. Once I have the results into my BLAST window, I can choose to either open up selected sequences with a specific Lasergene application using this drop-down menu, or I can add them directly into the alignment project. I can also launch the web browser, which would take me directly to NCBI's website for information pertaining specifically to that sequence. And then finally, I can batch save my results so that I can use them at a later time. Now, I do apologize for the delay in returning the results. With the webinar running, sometimes the connection to NCBI slows down a little bit. So once we have our results back, we can go ahead. Here we go. As you can see, I've got all of my BLAST results shown with the highest scores on top. These are the most relevant hits. And then I can actually see the hits down at the bottom with the information on how the alignment was done. Again, what I'm going to do is I'm going to add the top 10 hits directly to my alignment program by choosing the Add Project, and I'm going to save the top 10 sequences and click OK. And in the background, we can now see that all 10 of those sequences have been added, and I could perform an alignment.
Aaron, before you move on, we do have a question.
Sure.
Is it possible to edit the sequences that you've brought into Megaline? You showed how to set the ends. Are there other ways to perform edits to the sequences?
Yes. Yep. With the Megaline software, and I'll go into this a little bit more into detail once we have an alignment performed, but with the Megaline software, you can insert and delete gaps as needed to modify the alignment. However, you can't actually modify the sequence itself. To get around this, what we can do is select a sequence that we want to modify, and through a tool that's incorporated with the Lasergene software, we can choose File, Send Sequence to any of the other Lasergene applications. So in this case, I could send this file directly to SeqBuilder and make the modifications within SeqBuilder. And the nice feature, a part of this feature, is the fact that any sequence that we modify within SeqBuilder is directly impacted in the alignment. So you'll see the modifications immediately within the alignment within Megaline. So that would be how you modify the actual sequence itself.
Great, thank you.
Okay, moving forward, what I'm going to do is I'm going to enter in some of our sequences that I'm going to run an alignment on. And to do that, I went to the File, Enter Sequences, and navigated to this particular folder that contains some gamma-tubulin proteins. Now, to enter in the sequences, I can double-click on them and add them to the Selected Sequences window, or I can select on them and choose Add. So if there were just particular sequences that I wanted to add, I could highlight those and add those directly. Or, since all of the sequences are in this one folder and I want to add them all, I can simply select the Add All and add them to my selected sequences. I'll go ahead and click Done and enter in all of the sequences. Now this is what I'm going to do for this first alignment is one of our most popular Megaline applications, and that's the alignment of multiple protein sequences. Megaline does have the ability to perform multiple and pairwise alignments on both protein and DNA, but again, for this particular alignment, we're going to work with protein. Once we have our sequences entered, I can go to the Align menu, and I can show the various alignment algorithms that are used. The Megaline software has 4 methods for aligning sequences: the Jotunheim method, Clustal-V, and Clustal-W methods. The Clustal-W method actually has two methods to it: the fast approximate and the slow accurate. If you're not familiar with the various alignment algorithms, we do have information that within the help menu that can explain how each of the algorithms is going to be used for your data. However, just as a brief overview, the Clustal-V and the Clustal-W algorithms can be used for both nucleic acid and polypeptide sequences, and are strongly recommended when there's no reason to believe that the sequences are related by descent. Jotunheim, on the other hand, is recommended when the sequences are related by descent. So choosing the appropriate alignment algorithm is going to depend on your sequence data. Now, as I mentioned, I can go directly from the alignment menu and select the appropriate alignment method, or I can choose the method parameters and select the algorithm from there. In doing this, I have the option to adjust my parameters for the assembly, and there are a number of parameters for each of the algorithms that we include. However, we would always recommend that you use the default algorithms to begin, and then once you've done your alignment, if you need to make adjustments, you can do so. So again, we're going to choose the Clustal-V algorithm, and using the default parameters, I'm going to click Now, and go ahead and perform my alignment. Now, when the alignment first performs, you're going to see the work table view. And this is where you can examine the details of your consensus sequence at the top, as well as all of the component sequences below. The consensus is shown with a colored bar across the top, indicating how similar each of these sequences are to the consensus. And you'll also notice that there's a bar separating the beginning of the sequence to the end of the sequence. This bar just allows you to view various regions within your alignment so that you can get a couple of different views of all of your alignment. If you want to, you can move this all the way off to the side and just see a single view. Now, because the gamma tubulins in this particular alignment are very similar over the first 250 to 300 residues, you'll notice that, however, that the carboxyl termini are highly dissimilar. So what we can do is we can actually limit the size of the sequences over which to perform the alignment. And this might be helpful in phylogenetic analysis. So what I'm going to do is I'm going to just select the first 400 residues of this particular alignment by going to the Edit menu and go to Position. And now I can choose the position, which sequence I want to use. In this case, I'm going to choose the consensus, and I'm going to select a range from 1 to 415. I'm choosing 415 as opposed to 400 simply because I know that there are actually 15 gaps already in the consensus due to the assembly that we ran. So if I had just selected the first 400, it would actually only select 385 residues. I can go ahead and click OK, and as we can see here, we now have only the first 400 residues highlighted. It says 415 again due to those gaps. With those selected, I'm going to go into the Align, and I'm going to choose Create Alignment from Selection. And I'm going to go ahead and give this the name Gamma Tubes 400V, indicating that this is my alignment of gamma tubulins, only the 400 residues, and I've used the Clustal-V method. Go ahead and click Save, and it opens up a new alignment window with just those residues. From here, I'm going to run the alignment again and review our results. Now, the next view that I want to show you is once you have your sequences aligned, you can go into what's called our alignment report. And this report is very useful in publication or presentation of your alignment. You can modify the view of this report to customize it to exactly how you would like your report to look in a couple of different ways. We can go to the alignment report contents, and here we can show consensus, disagreement, or strength. We can modify how often the residues will break. We can choose if we want 3-letter versus single-letter codes. We can add additional rulers, and a number of other parameters that we can adjust. And once we have those set up the way that we want, we can go ahead and click OK. Another way to modify how the alignment report looks is by adding decorations to the alignment report. And decorations are a nice way to customize the report so that it's easier for you to evaluate disagreements between the various sequences that you've within your alignment. In this case, what I'm going to do is I'm going to hide all of the residues that match our consensus sequence. Now, I could also change any of the constituent sequences underneath and hide all of the residues that match, for instance, from human or from worm. But in this case, I just want to see all of the differences from the consensus. I'm going to go ahead and click OK. And as you can see now, anything that matches our consensus has disappeared, and we're left only with the variations. I can then create an additional decoration and say, shade all of the residues that differ from the consensus and further emphasize these differences. So I'll go ahead and choose a dark blue that should shade in everything that's the same, everything that's different from the consensus. And now we can very easily identify those areas of difference. Once you have a number of different methods applied to your alignment report, we can manage our decorations through the decoration manager. And this will basically allow us to modify the existing decoration or remove the decoration from the alignment report. So if I wanted to remove that last one, I could do so. Next, I'm going to go over the phylogenetic trees that Megaline creates. Megaline can give you a first look at possible phylogenetic relationships by displaying a preliminary phylogenetic tree. To display the tree, simply go into the View Phylogenetic Tree, and it will bring up the phylogenetic tree. There are a couple of different options for how you want to view the tree. For instance, if you wanted angled branches, you can click on the angled branches option on the left side, or if you wanted the straight branches, you can do so as well. Clicking the unbalanced tool displays the data as a cladogram in which the individual branch lengths reflect sequence distances. If you click the balance tool, the data is displayed as a phenogram in which the paired lengths are averaged. And as you can see here, we have some dotted lines in this view, and these dotted lines on the phenogram indicate a negative branch length, which is an artifact of averaging. So you can use the branch lengths when calculating the residue substitutions. You can further analyze your phylogenetic tree by applying bootstrapping to your results. The bootstrapping is basically a tool that is going to randomly sample the individual columns of the aligned amino acid sequence data from the original alignment. And the results describe basically how often a particular branch appears in a phylogenetic tree. So once we have our tree created, we can then go to the Align and choose Perform Bootstrapping. and this is going to bring up the parameters for our bootstrap analysis. There are two parameters, the number of trials and the random seed. And the number of trials basically indicates how many times the program is going to compare the display tree with random tree constructs using the neighbor joining methodology. And the higher the number that you use for the trials, the better your results. So we typically recommend using a range between 100 and 1,000. Now, the random seed is a tool that basically makes bootstrapping consistent with the Clustal interface. However, we would always recommend that you use the default random seed until you know more about how that's going to affect your results. But if you do have questions, again, clicking the question mark in the corner of the parameters box is going to bring you to the help menu so you can read more about how each of those parameters is going to affect your results. But once we have our results, we're going to click OK. And as you can see now, we have our bootstrapping scores at each of the branch points, giving an indication of the validity of this particular tree.
Aaron, we do have a couple of questions that have come up. One is regarding the alignment method parameters. Once you've adjusted those, is there a way to save those to use them again in future alignments?
If you have a blank window open with no sequence data entered in, so when you first enter Megaline, you can go into your method parameters and modify these parameters to however you want them modified. And then saving those will basically create global parameters. So anytime you enter sequences in, it will then use those new parameters. If you already have sequences applied, it's only going to apply these parameters to a particular project. So you would have to, in that case, modify the parameters each time you enter in sequences. But again, these parameters are what are recommended within the papers. So modifying them isn't something that you would necessarily want to do to all of your alignments. It is good practice to manually adjust the parameters so that you know exactly how each of the alignments is going to be affected.
Thank you. And then another question, is there a way to export, say, the phylogenetic tree or the alignment report to use in a publication or another application?
Absolutely. It's very simple to get the data out of the program. It's simply selecting, going to the Edit, Select All, and that's going to select the tree, and then just simply copying and pasting into the application of choice. For the phylogenetic tree, it's going to copy and paste the entire tree. For the alignment report, you can select individual rasters, or again, you can select the entire thing and copy and paste that. But these images paste very well into Microsoft Word, PowerPoint, or other programs that can display the images.
Great. And just one more question. Is it possible to blast the consensus sequence to look for other more distant homologs after you performed your alignment?
Absolutely. It would be the exact same thing that we did in the initial entering. You'd simply select the consensus and go to the net search and choose blast selection. And that will bring your results back in the similar fashion as the earlier search that we performed.
Great. Thank you.
Okay. So with those views shown, we do have a couple of other views where you can view residue distance and residue substitutions tables, but they're not quite as interesting as the tree or the alignment report. So I'm going to go ahead and skip those for now. With that said, however, what I do want to show you is variations in how the algorithms deal with the variations within the alignment. As you can see here, for the Clustal V algorithm, we have an area of variance right around the 290 mark here. And I've actually performed an alignment using the Clustal W slow accurate method with this exact same data set. So really quickly, I'm going to go ahead and open that particular alignment, and I'm going to scroll to the same area so that we can see how the two different algorithms have effectively handled this data. Actually, that's the wrong alignment. So let me back up here. What I'm going to do is I'm going to do a file save as, and we'll go ahead and name this one clustLW. Save, and rerun the alignment. And now I'm going to open up the clustLV alignment that I did. And we'll scroll to that region. Oops. There we go. So this region right here. And then we'll go to the clustLW alignment and go to the same region. And why isn't it? Well, I'm having a little technical difficulty in opening up the actual alignment. But you would notice that there would be differences in how those alignments were performed. So again, it's always a good thing to make sure that you choose the appropriate alignment algorithm for your particular data set. And then with that done, just really briefly, I'm going to go over the pairwise alignment. And to do this, I can either enter in just two sequences into my alignment, or I can select two sequences from a particular alignment that I've already performed. In this case, I can use, say, the sea urchin and the worm. And I'll go ahead and from the alignment method, or from the alignment menu, I'll select one pair. Now we offer three different alignment algorithms for our pairwise alignments. We use the Wilbur-Lippmann method, the Martinez-Needleman-Wundt method, and the Lippmann-Pearson method. We also offer a dot plot for the pairwise alignments. Now the algorithms know that because this is a protein alignment, the only algorithm that is usable for proteins are the Lippmann-Pearson or the dot plot. If we were to have DNA, the Wilbur-Lippmann and the Martinez-Needleman-Wundt methods would also be active, and the Lippmann-Pearson would not be. So in this case, I'm going to go ahead and choose Align One Pair by the Lippmann-Pearson method. Again, it brings up my parameters that I can adjust, and click OK. And here is my pairwise alignment. Similar to the alignment report, we can modify how this particular alignment is viewed. By clicking on the alignment view options, we can bring up our parameters for color. So if I wanted to say show everything that mismatches in red, and instead of the residue character, I want to see a vertical bar. I can also add names to the alignment. I can remove the header or the rulers. And I can also show context of the alignment. So if, for instance, you've just selected a section of your alignment, it will extend that selection so that you can see the where that particular sequences are located within the overall alignment. And again, as with the phylogenetic tree and the alignment report, I can select this and put this into a Microsoft Word or PowerPoint document. So that's going to wrap up the Megaline section of the webinar.
Aaron, there's one more question regarding Megaline before you move on.
Yes.
In your example with the clustLV and clustLW alignment, you cut off your sequences around base 415. How would someone identify what the best place or range of sequence to use is?
Well, that's basically deals with a bit of knowledge within your sequences. In this case, we know that these particular sequences have a high degree of variability at the ends, which can really impact phylogeny. I just selected the first 400 bases because I knew that the similarity was a bit stronger within that region. I could have done the phylogenetic analysis based on the entire assembly, but it would have modified the phylogenetic results. And again, with anything that you do for assembly purposes, whether it's selecting a region that you want to align or selecting the algorithm that's aligned, these are decisions that you as an individual researcher will have to make and justify. So for making a tree that looked a little bit better, we selected those 400, the first 400 residues. If you're not sure, we would always recommend just using the entire sequence information for your trees. Just for justification purposes.
Great, thank you.
Mm-hmm. So with that done, I'm going to go ahead and close up the Megaline application, and I will go into the GeneQuest application. So basically, GeneQuest is going to aid you in identifying and annotating coding regions and other features of interest in your DNA. The project is going to begin by entering in a sequence that you want to analyze. Oops. And so I'm going to choose New, and I'm just going to enter in my DNA sequence. Now, by default, when you first enter in a sequence into the program, you're going to see three windows. You're going to see two panels, or curtains as we call them. This is the method curtain on the left, and here we have our legend curtain on the right. Now, in the center is our assay table, and the assay table is basically going to show you all of the methods that you've applied to your sequence. And I'll explain exactly what each of the methods do for your sequence. But just real briefly, by default, the outline of what you're going to see in your assay table is a scale representing the scale of your sequence. This black bar, which is actually the sequence, it's just that we're zoomed out so far that it's represented as a solid black bar. If I were to actually select a region to zoom in, you could then see that particular sequence. But we're going to zoom back out just to give us a better view of our overall sequence. By default, we also have some open reading frames applied, as well as some start codons that are associated with the sequence. To apply additional methods to GeneQuest, simply go to the methods curtain and select the method of interest. If the method that you're interested in isn't shown, or if you would like to see the same method with different parameters, you can simply click on the More Methods and add additional methods to the curtain. In this case, if I wanted to add some starts and stops, say I wanted to show the stop codons for this particular sequence, I can select the first three reading frames and drag them directly into my assay table. Now you'll notice that they have all shown up as red. By default, whatever the last color that you set to your methods is going to be the color that's applied to any methods that you add from here. We can easily modify the color of each method simply by right-clicking on the method and going to line color and then say changing it. We can also stack individual methods. So if, for instance, I just wanted to see all of our starts in one particular line, I can do so simply by stacking those. You'll notice that what we've done here is we've identified each reading frame by a color, so it's easy to see which frame corresponds to each reading frame by keeping this color scheme consistent. And that'll be very important when we get into the next project that is going to show you in detail how to go about discovering your genes. So with that said, I'm going to go into a program or a file that's already been created. And now there's a bit more information that is on this particular sequence that wasn't on the last one. In particular, what we've done is applied some tools that are very helpful in locating potential coding regions. And I'm just going to scroll to a specific region here, and then I'll explain. What we have here are a list of acceptor sites and donor sites, as well as Borodovsky matrices. And for those of you who aren't familiar with Borodovsky plots, these plots are used to evaluate coding potential independent of identifying open reading frames. So combining Borodovsky plots along with open reading frames gives you a very accurate way of identifying potential coding regions. Then utilizing the start sites or acceptor and donor sites, you can then begin to identify the coding regions of interest. Again, as you see, we've got the reading frames color coordinated, so it's very easy to see. We have frame 1 in red, frame 2 in green, and frame 3 in blue. So we can very easily identify when we have a Borodovsky peak inside a particular reading frame. Now the next thing I wanted to just point out is each of these matrix for acceptor sites and donor sites and the Borodovsky matrices are specific to the C. elegans. The GeneQuest module comes preloaded with a number of matrix files that are very specific to various organisms. So we would always recommend that you use a matrix file that is the organism that you're working with. And if we don't supply that particular organism, we would recommend that you select an organism that is as closely related as possible, because the higher the relationship, the more accurate your results will be. So to begin with the identification of coding regions, we're going to scroll to a region that we can see some correlation between Borodovsky peaks and open reading frames. And in this case, we have here a open reading frame in the third frame that corresponds to a Borodovsky peak, also in the third frame. So this is going to be where we'll start with our annotation. The first thing you need to do is identify a potential start codon. So we have our start codons applied at the bottom here, and we have 3 that fit within this particular open reading frame. And if I click and hover over each of these, it pops up a score. In this case, we have a 9, we have a 6.6, and we have a 3.7. You can also see these numbers by zooming in a little bit closer. Now this 9 here is a very good candidate for our start site, because it's not only within the open reading frame, but it is also at the beginning of the Borodovsky peak. So this is most likely going to be the start site of our first gene. From here, we need to identify if there's either a stop site, or if this is a multi-exon gene, we need to identify a donor site. So being that the open reading frame ends over here and our Borodovsky peak ends considerably earlier, and we do have 2 donor sites that we've located. It's most likely that this is just the beginning of exon 1 here. So we have 2 scores for our donor sites. We have a 9.6 and a 4.6, and again, the higher the score, the more likely the candidate. So in this case, I'm going to hold down the Shift key, click on our donor site, and select the region that will become exon 1 of our first gene. Once the section is highlighted, I'm going to use our annotation tool on the left-hand side, and this brings up our annotation box where I can then give our gene a name, and I can give our section a name, exon 1, and I'll go ahead and click OK, and we've now annotated exon 1 of our gene. So moving forward, what we need to do is identify another Borodovsky region, and in this case, we don't need a start site. What we need is an acceptor site. So we can see that we have a nice Borodovsky peak starting, and we have an acceptor site with a score of 12. So that's a very high score for acceptor sites. So I'm going to zoom out here just to make it a little easier. And this is very likely a candidate for the beginning of exon 2. So I can click on exon 2 and scroll to the end of our Borodovsky region. Again, this is in Open Reading Frame 2, so you can see the color coordinating with the ORF. We now then find our donor site with a score of 12.6. Again, this is a very high score, so it's very likely the donor site for this particular exon. So I'm going to hold down the Shift key once again, select our donor site to highlight the region, and now instead of selecting the annotation tool, I'm going to click the joining tool. And the joining tool allows us to add this particular segment to an existing annotation. So in this case, gene 1 already has exon 1, so I'm going to join it to that. And if I zoom out just a little bit, we can now see that exon 1 is joined with exon 2, and they're all listed as gene 1. Moving forward, we have another peak right here. So we need to locate our acceptor site, and we have a candidate site right here. Now you'll notice that not only is this next Borodovsky peak in the same frame, but it's also in the same open reading frame. So just a little bit of information there. We're going to go ahead and select the donor site, the acceptor site at 14.1. And as we move to the end of this Borodovsky peak, we can see that we have 2 donor sites. We have a 6.4 and a 4.7. So most likely the 6.4 with the higher score is the best candidate for our donor site. Go ahead and select that. Choose the linking tool to gene 1. And we have our third exon annotated. And continuing down, we now see one more peak, again in the not only in the second frame, but in the same open reading frame as exons 2 and 3. We look for our acceptor site. In this case, we see one right here with a score of 15.1. And we go to the end of our Borodovsky peak, and there really aren't any donor sites here. And we also see the end of our open reading frame. So this may indicate that we've actually reached the end of our gene. To validate this, I'm going to go into the coding prediction and apply the second reading frame stop sites. And as you can see, we actually do have a stop site located right at the end of our open reading frame. And this is most likely then going to be the end of our gene. So I'll go ahead and select the acceptor site, hold down the Shift key, and select our stop site, and go ahead and link this to our other exons. Now, to complete the annotation, I've double-clicked on the gene that we created, and as you can see here, we've got gene 1 with exon 1 annotated, but we don't have exon 2, so I'm just going to go ahead and give that a title. And exon 3. Oops. And then exon 4. Go ahead and click OK. And we now have a fully annotated coding region. The next step in coding, in locating potential coding regions, is basically going to be identifying or verifying that we have identified a potential coding region here. And to do this, all we're going to do is highlight the entire gene, and we're going to choose the Net Search menu, Blast ORF. So what we're going to do is send this directly to NCBI and see if we have any similarity with known annotations of organisms. So we'll just wait for the results to come back.
Aaron, while we're waiting for that, a couple questions. One actually very related to what you're showing now, but wondering, is it possible to blast against the protein database for your annotated exons?
Yeah, you can. It is possible to select the database that you want to blast to, and I'll show you that in just a second here. But with our results found, just really briefly, what the easiest thing to do would be to take the top hit, because that's the most likely candidate for a potential match, and we're just going to go ahead and put that into the application here. So I'm going to click OK, and we now have this annotated region, and as you can see, it lines up nearly perfectly with the annotated region that we discovered. So as Katie had asked, if we just wanted to blast a protein, we can go to the blast selection and choose blast P, blast X, T-blast N, or T-blast X, depending on which database we want to blast to. We can also select the specific database that we want to blast against. And if you have any questions on which program or what program each one's blast to, again, click the question mark and there's a description. So again, in this case, we are blasting nucleotide to, well, actually, we could blast P this and it will translate it. We could also blast the nucleotide information and have it translate to a protein, or show the 6, in this case, if we chose T-blax-X, we could search the 6 reading frame translation with the 6 frame translation of a nucleic query sequence. So depending on the program that you choose, you can blast the protein. So that really goes through the annotating of a potential coding region. And the one last thing that I wanted to show briefly is, say you have a list of your own exons in a file, and you want to just see where those line up on a particular sequence. What we can do is enter in the sequence that you want to search against, and using our gene finding tool, we'll choose DNA Finder, and in this case I have 2 sequences that represent exons, and I'm going to select those and click Done. Now they've appeared down in my More Methods, and I now have the option of adding the top strand or the bottom strand for either of the exons, or a composite of both of them. So in this case, I'll grab the top and the bottom strand composite for those particular exons, add them to my assay, and then I'll just zoom out so we can see where we've found some hits. So as you can see, we have a number of hits across the bottom here, and each of those numbers is representing how many residues actually match at that particular region. So if we zoom in close enough, we can actually see the sequence that matches. And you can do that with all sorts of DNA. So if you also have a protein that you want to search against, you can use the gene finding protein finder as well. So again, that covers the various workflows that we wanted to show you today for both the Megaline and the GeneQuest applications. So at this point, I'll hand it back over to Katie.
Yeah, we actually have a lot of questions coming in, and we have just a few minutes, so I'll ask as many of them as we have time for. But again, we can follow up with people later. One just general question about GeneQuest. Can you sort of talk a little bit about what kind of input files you can give GeneQuest?
As far as
like what?
Oh,
sorry,
sure.
What kind of files you can open in GeneQuest?
So really, GeneQuest can handle both. Well, it's going to be a DNA sequence that you want to enter into the program. As far as the file types, you can enter in a number of readable files. So if you have sequence directly from NCBI or within a particular Lasergene application, you can use trace data. But almost any file that has DNA information in it can be opened within GeneQuest. The most interesting files that you're going to want to use are going to be larger unknown sequences. With the amount of information available at NCBI, locating novel sequences is becoming less and less, or easier and easier to do with some of the other tools. So this is really designed if you have a novel strand of DNA and you're not really sure where to begin looking for those particular coding regions.
Okay, and like I said, we have a lot of questions, so I'm going to do a couple more. One, how much memory do you need to run Lasergene or specifically GeneQuest on a desktop computer?
Sure. It's really not that much in actuality, and what I'll do is I'm going to just bring you to the DNASTAR homepage for specific information. So if we go into the Lasergene Core Suite, there's information on the technical requirements right here on our web page. So we do require for Windows XP or Vista or Windows 7, we recommend about a gigabyte of RAM and about 400 megabytes of hard disk space. So not very much as far as the technical requirements. However, if you're using larger files, especially in GeneQuest, if you have, say, an entire genome that you're using, obviously the more RAM, the better. So it will perform better with more RAM.
Great, thank you. I'm actually going to wrap it up there, but I just want to reiterate that we will follow up with anyone whose questions we didn't get to after the webinar. We also will post a recording of this webinar on our webinars page, and we'll also be posting some new webinars in the next couple weeks. So check back if you want to see more of these live demonstrations. We should have some more webinars at the end of this month and into the fall. And we also have a large collection of videos. We have over 100 videos, each about 3 to 4 minutes long, showing some of these workflows in depth. So if you want to see more about gene finding or alignment or any of our other applications, our video library is a great resource for that.